So let me briefly explain to you what the DNA uptake process in Vibrio cholerae looks like. So under the right set of conditions, Vibrio cholerae will produce this competence apparatus, which is composed mainly of subunits of pidins, primarily pil A. It extends out into the environment and contacts DNA, and then by a process of depolymerization, DNA is pulled in. Once DNA is pulled in, it's protected by the DNA binding protein COMA, which helps it shuttle into the cytoplasm through the inner membrane channel of COMA. Once DNA is taken up, it can be used for nutrition, so just as a food source, for double-strand break repair. So if there's a break, bacteria don't have homologous end-joining mechanisms, non-homologous end-joining mechanisms. So they have to rely on double-stranded piece of DNA to repair any double-strand breaks. And finally, it can undergo genetic exchange and natural transformation. I should also mention that this is not a constitutive phenotype, but it's activated under a very specific set of conditions. So what are these conditions? One set of inputs is from chitin colonization. So presence of chitin leads to upregulation of a transcriptional regulator called TFOX. TFOX in turn activates another transcriptional regulator called Questor, and it's thought that and TFOX can control Questor as well as many complement genes. Quorum sensing is another input. So when cholera reaches high cell densities on chitin, it activates a transcriptional regulator HAPR, which also functions through Questor. And Elena, who was my predecessor in the lab, she discovered another input called CyTAR, which is activated when these cells are starved for nucleotides. And so what I was interested in when I joined the lab is trying to figure out how does CyTAR link with this regulatory scheme. So I came up with the simplest model, which is all of these transcriptional regulators eventually end up at Questor, and then everything is funneled through Questor for competence. So in order to test this, I did the following assay. So this is just a reporter assay. What we've done is fuse the Questor gene promoter to an operon that codes for luciferase. And so whenever this gene is being expressed, or when it's being transcribed, the output is light units. So we can measure light production. And as you can see, in wild type cells, the Questor expression is high. If you knock out either TFOX, HAPR, or CyTAR, it goes down, which indicates that all these three are necessary for activating Questor expression. And so, so that told us that CyTAR activates Questor. So the simplest model seems to be right. But if that's the case, and if we overexpress Questor and get rid of everything else, the expectation would be that this would be sufficient to activate competence and natural transformation. So let's test that hypothesis. The way we do this in the lab is we grow our Vibrio cholerae cells on a piece of crab or crab shells. We give them antibiotic marked genomic DNA or PCR products. We plate on selective media, and then we enumerate transformation frequency as the number of antibiotic resistant CFUs divided by the total CFUs. And this is what we see. So when Questor is overexpressed, you see that the transformation frequency is high. If you knock out HAPR, they can still transform, but not to the same extent. But if you knock out TFOX or CyTAR, then Questor overexpression is no longer sufficient to activate transformation. So this indicates that a simple model is not the whole picture, that there are other inputs from TFOX and CyTAR into competence. So what are these inputs? So in order to figure this out, I did, with the help of Jacob in the lab, an RNA-seq experiment with strains that were induced for competence that were either deleted for TFOX or deleted for CyTAR. And so what I'm showing you here is a heat map of competence genes that were activated by TFOX and CyTAR. So columns 2 and 4. So column 2 corresponds to genes that are upregulated by CyTAR. So yellow here means upregulation. And column 4 points to competence genes that are upregulated in the presence of TFOX. And you can see that the pattern is roughly similar. So they seem to coordinately regulate competence genes. And with further reporter-based analysis, I was able to show that the regulation of competence genes is more complex. That competence genes can be subdivided into 4 different classes. Class 1 is dependent on the presence of all 4 transcriptional regulators. So you can see that their expression goes down if you knock out any of these 4. Class 2 requires only TFOX or CyTAR. Class 3 requires only TFOX. and class 4 doesn't seem to require inputs from any of these regulators. And this scheme of regulation, if you overlay it with the competence apparatus, seems to indicate that competence and transformation is actually a two-step process. Most of the prilin genes are controlled by TFOX and CyDAR, but only when they reach high cell density and they're expressing HAPA and Questor is this periplasmic DNA binding protein produced. And so that means that only at high cell density are they able to take up DNA into the cytoplasm. So this pilus is made by TFOX and CyDAR, but they require high cell density for taking DNA out and undergoing transformation. So we conclude from this that CyDAR and TFOX control multiple competence and transformation genes, including Questor itself. In addition, using the RNA-seq data set, we were able to see that CyDAR and TFOX also regulate multiple extracellular kinases that Vibrio produces. So you can see the pattern here that in all 4 cases, if you knock out TFOX and CyDAR, the expression of kinases goes down. And similarly, if you knock out CyDAR or TFOX, their ability to degrade chitin and produce this zone of clearing disappears. And they seem to produce these fine if you knock out HAPA or Questor. So this indicates that CyDAR and TFOX control multiple kinases, but that regulation is independent of HAPA and Questor. And finally, to our surprise, we also saw that CyDAR and TFOX positively regulate genes for a type 6 secretion apparatus. So as I mentioned earlier, the type 6 secretion apparatus is an apparatus that Vibrio uses to stab its neighboring cells and deliver toxic effectors into neighboring cells that sometimes leads to cell lysis. These cells are protected from the toxic effects of the effector by these immunity proteins, by these cognate immunity proteins that protect against the toxic effects of the effector. So they are protected by attacks, by self-toxicity, as well as attacks from sister cells, but they can kill cells that don't have these immunity proteins.